Alright guys, we've made it to our last lecture. Our last lecture is going to be on reproduction. There's a lot more to this chapter than we're actually going to cover, mainly because it's the end of the semester and you guys have a lot to prepare for on your finals, and so we're going to just cover a few key topics of this chapter. So let's look at the functions of the reproductive system. One is the reproductive system produces sex hormones that are going to influence growth and development of organs and tissues. The reproductive organs also produce gametes, and gametes are the cells such as sperm in males and ova or eggs in females. For the reproductive organs, in males, the gonads are the testes, and the testes produce the gamete, which is the sperm. And in females, the female gonad is the ovaries, and the ovaries produce the gamete for females, which is the egg or ova. And then the other purpose of the reproductive system, and this is the main purpose, is actual reproduction. So when the sperm and egg come together, the goal is to produce offspring. So the male's reproductive system is designed to deliver the sperm to the female reproductive tract, and the female's job is to provide an environment for the fetus to develop. So let's look at the human life cycle a bit, because this will help you understand the production of the gametes, the sperm and the egg. Okay, and you're going to have to review two terms. Remember the term from when we learned mitosis, you learned the term diploid. And diploid means that each cell contains two sets of chromosomes. So we actually have 23 types of chromosomes, 2 of each. So we have a total in our cells of 46 total chromosomes, 23 different ones. So in humans, all your cells have 46 chromosomes for the most part. And so in our gonads, in the ovaries and the testes, we have stem cells that have 46 chromosomes. Now those stem cells are going to undergo a type of division called meiosis. And this type of cell division basically cuts the number of chromosomes in half. So these stem cells will divide in the testes and the ovaries to produce the gametes, which are haploid. They only have one of each chromosome. So they have 23 total chromosomes. Okay, so they only have one set. So what happens is the ovaries and the testes, the stem cells undergo the type of cell division called meiosis to produce gametes, which are haploid. And then those two gametes will come together through a process called fertilization. And so during fertilization, the sperm contributes 23 chromosomes and the egg contributes 23 chromosomes. So a set of chromosomes from each. So you get back to having two sets and being a diploid zygote. Okay, so a zygote is basically a fertilized egg. And then what will happen is that cell, the zygote, will undergo cell division, but this time it's going to undergo mitosis, because you learned about in mitosis the cell divides and the resulting cells also have 46 chromosomes. So that cell will divide into 2 and then you get 4, and the cells keep dividing until you actually end up back as a multicellular diploid organism. Okay, and so that's kind of the little life cycle. Now if we look at our chromosomes again, you have 23 different types of chromosomes, 2 of each, in the stem cells that are going to produce the gametes. Okay, and the 23rd pair is what we call your sex chromosomes. So in females, if you're a female, your stem cells in your ovaries have 2 X chromosomes, your XX. And if you're male, the stem cells in your testes have an X and a Y. Now these cells are going to produce the gametes through the process of meiosis. And in meiosis, you got to cut the number of chromosomes in half. So in meiosis, you actually only donate one of these chromosomes to the cell instead of both like you would in mitosis. Females will produce eggs that either have an X chromosome or an X chromosome, because that's the only chromosomes they have. So in females, all eggs have an X chromosome. In males, though, they can produce sperm that either have an X chromosome or some sperm that have a Y chromosome, because only one goes into each cell. And then you have fertilization. So if you have an egg that gets fertilized with a sperm that's carrying an X chromosome, then your offspring will be female. It will be XX. If you have an egg that is fertilized by a Y chromosome, then your offspring will be male, XY. So it's actually the sperm that determines the sex of the offspring. I always kind of find this a little humorous in that in history, males would want to have a male offspring because back then males inherited the wealth and land and different things, and so it was very important to have a male offspring. And a lot of times in history, kings and so forth would either go get a new wife because their wife wasn't giving them a male offspring, so they kind of thought, oh, I need a new wife. It was the wife's fault, but it's actually the sperm that determines it. Alright, so let's talk a little bit about once you have fertilization, the embryo starts to develop and is dividing through mitosis. And during development, whether you're male or female offspring, during the first 6 weeks, we all look the same. Okay? Our internal reproductive organs look exactly alike, and we all have the same tissues. And these tissues, we say, have the potential to develop either into male anatomy or female anatomy and physiology. And so we call these tissues bipotential tissues because they have the ability to become either or. And so we all had these 3 general tissues. We have a bipotential gonad, and this gonad could either become later on the testes or the ovaries. And then you have 2 ducts. We all had a Wolferian duct and a Mullerian duct, okay, up until 6 weeks. But then what happens at 6 weeks is the males, if you're XY, the Wolferian duct will develop into your male anatomy, and we'll talk about that, and the Mullerian duct disappears. If you're female, the Mullerian duct remains and the Wolferian duct disappears. Okay? But we all had both of these at one time. What makes the difference? What causes the change to happen after 6 weeks? Well, it all has to do with if you have a Y chromosome or not. So if you're male, you have the Y chromosome, and there's a gene that is found on the Y chromosome called the SRY gene. It's called the sex region of the Y chromosome. Okay, sex determining region of the Y chromosome. And this SRY gene, if it's there, through transcription, it will start to produce what is called the SRY protein. Okay, and the SRY protein will then turn on other genes, and you basically are turning on genes and making certain proteins which will cause the bipotential gonad to start to differentiate into the testes. And so the medulla of the gonad, the center of the gonad, will become the testes, and the cells will start to differentiate and start to have certain jobs. And 2 types of cells that differentiate in the testes are the Leydig cells and the Sertoli cells. And once they differentiate, they'll start to produce additional hormones. And the Leydig cells will produce testosterone. And what testosterone will do is it will cause the Wolferian duct to start to develop into male anatomy. And then testosterone, along with another hormone which is very, very similar to testosterone, it's called dihydrotestosterone or DHT. DHT is very important in the development of the external genitalia of male. And this is kind of interesting because genetically you can be male, you can be XY, but there's some things that can happen during development, and there's a Y sex-linked disorder, genetic disorder, where the gene that produces the protein receptor for DHT doesn't function properly. So there's some people that genetically don't make the receptor for DHT. So even though genetically they're male, XY, and they might produce DHT, it doesn't bind to the receptor. And so what happens is the external male genitalia doesn't develop normally. And so when they're born, anatomically, externally, they can look female. Internally and genetically, they could be male. What used to happen is when you kind of had these questionable external anatomy, doctors would just say, well, looks pretty much female, not quite sure, just raise them as a female and they'll be fine. Well, now we've kind of learned that that doesn't work very well, okay, because that can cause children a lot of problems if genetically they are male, but you're trying to raise them as a female. And so now, a lot of times they'll do a genetic test and they can see chromosomal that they're X or Y. But that's just kind of an interesting story that can happen. There was also a family in South America that this is how it was kind of determined. Their external genitalia developed as female, because female is the default if these hormones don't work properly. So they were raised as female. But what then happened is when they hit puberty and they started to ramp up their testosterone production, they started to develop secondary characteristics as a male. Because genetically they were male, just externally they looked female. And so they were raised female, and then all of a sudden they were getting male secondary characteristics because the testosterone started to be produced because they do have the Y chromosome. They were getting deep voices and so forth. And it ran in a family. So it was a genetic abnormality. Now, the Sertoli cells produce what we call the anti-Müllerian hormone, which will cause the Müllerian duct to disappear. So that's what causes the Müllerian duct to disappear, and the Wolferian duct remains. Now, in females, if the Y chromosome isn't there, you don't have the SRY gene, so you don't produce the SRY protein. So in the absence of the SRY protein, that's what I kind of mean by default. If the protein isn't being produced, then the gonadal cortex, the outside of the gonad, will differentiate into the ovaries. And then the Wolferian duct will degenerate in the absence of testosterone because testosterone's not there, and the Müllerian duct will remain because you don't have the anti-Müllerian hormone. The Müllerian duct will become the female anatomy, such as the fallopian tubes, uterus, and the top part of the vagina. There is also another disorder, I'm just thinking of this, because it's something to be, aware of, is a lot of these anatomical genetic things have to do with genes not functioning properly. You could be XY, but for some reason your SRY gene isn't producing the protein properly, so anatomically you could develop as a female, but genetically be male. So this is what it kind of looks like. Bipotential stage. So for the first 6 weeks of fetal development, we all look the same. You have a bipotential gonad, you have the Wolferian duct, which is attached to the gonad, and then the Müllerian duct here. And then what happens is, if you're male, about that time, you start to produce the SRY protein, which causes the medulla to differentiate into the testes. And once the testes have differentiated, the Leydig cells will start to produce testosterone. And testosterone will cause that Wolferian duct to develop into basically the seminal vesicles and the vas deferens, which is the tube that leads from the testes and then to the urethra. And then the anti-Müllerian duct will cause this Müllerian duct to just degenerate and disappear. Now, if you're female and you don't have the SRY protein because you don't have the Y chromosome, the cortex of the gonad will become the ovaries, and the Wolferian duct will disappear. And then this Müllerian duct here will start to develop into the fallopian tube, and that's why the fallopian tube isn't actually physically attached to the ovary, and you still have remnants of the Wolferian duct. You have ligaments that attach to the ovary that hold them to the posterior abdominal wall. That's a remnant of the Wolferian duct. The Müllerian duct will become the fallopian tubes, the uterus, and the upper part of the vagina. Now, it kind of goes the same for external genitalia. So that's all internal anatomy. External genitalia, you also have bipotential structures, basically, that look identical at the beginning of development, and then they'll differentiate. You have a bipotential structure called the genital tubercle that's the same in both male and female, but what will happen is in females it will become the clitoris, and in males it will become the glands of the penis. And then you have urethral groove, which is a bipotential structure. It's the same in both, but it will become basically the labia minora and majora in females, and in males it will become the shaft of the penis and the scrotum. And it's the DHT that causes the development of the male external genitalia. So if you don't have, for some reason genetically you have the Y chromosome but you're not producing DHT properly because that gene's not functioning, or you don't have the receptors for DHT, just in the absence of those hormones, even though you're XY, you can develop as female. Anatomically it looks like you're a female. Oh, and I should mention the testes, they develop in the abdominal cavity, and then they have to descend into the scrotum, which happens later during development. And that could not happen properly also, where the testes don't completely descend. And this is just a summary of what the bipotential structures are that we all have, and then what it becomes in males and what it would become in females. So next we're going to talk about how the gonads, either testes or ovaries, produce the gametes, the egg and the sperm. And it's done through a process of cell division called meiosis. So you've already learned about mitosis. Now we're going to learn about meiosis. But let's first review our chromosomes. And you learned this when we learned mitosis, but that was long ago, back in our first unit. So I don't think it will hurt to review some of the terms. But remember, in humans, you have 23 pairs of chromosomes. So for each type of chromosome, you have 2 chromosomes. And each somatic cell has 2 sets of chromosomes, one maternal and one paternal. So it's said to be diploid. And the stem cells in the gonads are diploid. You guys don't get much genetics, Mendelian genetics, or inheritance. So we're going to briefly give you some idea of how you get the traits you get, but this is very brief. So remember, we call these homologous chromosomes, and they carry equivalent genes. So this chromosome 1 carries the same types of genes as this chromosome 1. Okay, so it might code for eye color, hair color, so forth. They both carry the hair gene, let's say. But they might carry alternate forms of each gene. So one might carry blonde hair, the other one might carry brown hair. Because you got one of these from the sperm, from your father, and one of them from the egg that came from your mother. And those alternate forms we call alleles. Okay. Now your gametes that you're going to produce, when you produce egg or sperm from your stem cells, they're only going to get one of each of these chromosomes. So just one chromosome 1 goes into an egg, the other chromosome 1 will go into a different egg. And that goes for each of these chromosomes. So you're only going to put one chromosome 1, one chromosome 2, one chromosome 3 into each egg, and that egg would be haploid because it only got one set of the chromosomes. And we talked about, like, you'd only put, if you're male, you'll only put one of your sex chromosomes. You'll either put an X or you'll put a Y. So what happens is when you produce eggs or sperm, you're only giving half of your genetic material to that egg. The other half is going to come from the other gamete that fertilizes it. So the purpose of meiosis, it's a different type of cell division, because in mitosis you pass all the genetic material, you make a copy of all your chromosomes, and the new cell gets each of those chromosomes. Now in meiosis, you're only passing on one of the chromosomes. So meiosis is a type of cell division that's used to produce the gametes, and sperm and ova are the only human cells that are going to be haploid. And the purpose of meiosis is to reduce the original number of chromosomes in half, because you only want to put 23 in there because you're preparing for it to fuse with another gamete who's going to donate the other 23. If you didn't reduce the number of chromosomes in half, every time you had fertilization, basically the chromosome number would double. And after many generations, we'd have way too many chromosomes. The other thing that's unique about meiosis is you only pass on a unique set of chromosomes in each gamete. Okay, and we'll talk about that because you're only going to pass on one of each pair. And so we say you're scrambling the parent's genome, okay, and that's called recombination. So let's look at meiosis, and I'm going to kind of go through this pretty quickly. It's similar to mitosis, so I'm just going to kind of point out the differences because there is differences. The overall purpose is different, and what you're seeing here is a general overview. So you're only looking at one homologous pair. So you have to think in humans you have 23 of these, but they're just showing what's happening with one set. So this would be the stem cell in your ovaries. You have one that came from the sperm and one that came from the egg when you were conceived. And what will happen is you duplicate these into sisters. So now you have them duplicated into sisters, and this is called a homologous pair. And when they're duplicated like this, we call this a tetrad. And what will happen in the first meiosis division is you separate the homologous chromosomes. So this duplicated chromosome will go to one cell. This duplicated one will go to the other. Sorry, after the first division, you already have 23. But the deal is these 23 are still duplicated. So what I mean by that is you're separating the homologous pairs. So if you can imagine that you have 23 of these line up, so 23 line up here, 23 line up on this side, and then you separate them in the first division. These 23 go to one cell, these 23 go to the other cell. So you still only have 23 chromosomes, but they're still in sisters. They're duplicated. And then in the second division, they line up, and you separate the sisters. Okay, so this sister will go one way, this sister will go the other. So you still end up with 23 because you only have 23 duplicated ones that line up, and then 23 of the sisters go this way, 23 of the sisters go that way, and you end up with cells with 23 chromosomes each. So to summarize, in the first round, you're separating the duplicated homologous chromosomes. So the homologous chromosomes separate. In the second round, you're actually separating the sister chromatids. So let's go through this. In interphase, it's the same. You guys already learned about interphase. Basically, your chromosomes in interphase during S phase, remember the S phase, you duplicate all your chromosomes into sisters. Then we go through meiosis 1. So meiosis 1, you go through prophase 1, metaphase 1, anaphase 1, telophase 1, and then you do your second division, which we call meiosis 2, and you go through prophase 2, anaphase, or metaphase 2, anaphase 2, telophase 2. So in prophase 1, what happens is your homologous chromosomes pair up in tetrads. Okay. So they find each other. So this would be chromosome 1, and let's say this is chromosome 2 and chromosome 2. But they're duplicated into sisters. Now what can happen during this time is you can swap pieces of your homologous chromosomes, and this is called crossing over, because these are the same chromosomes, so they carry the same types of genes, but remember they may carry alternative forms. So let's say this paternal chromosome they both carry the hair color gene, but one's carrying blonde and one's carrying brown. Well, what can happen is when they touch, they can swap. So now what ends up happening is you have a chromosome that has a combination. It has some of the maternal, it might have the maternal hair color, but then it has the rest of the genes on that chromosome are paternal. Now this has to occur between non-sister chromatids. Why? How are non-sister chromatids different from sister chromatids? Well, remember, sisters are identical to each other. So it wouldn't matter if you swapped the hair color gene between these two sisters here, because they'd both be blonde. So it only really happens between non-sisters, which would be these two. Now in metaphase 1, this is a little different than mitosis. Remember in mitosis, if I had my cell here, all the chromosomes lined up in a single file line. So you would have 46 of them lined up in a single file line, and they're duplicated. So what would then happen is 46 sisters go this way, 46 sisters go that way, and you end up with two cells that have 46. That was in mitosis. In meiosis, in metaphase 1, they line up with their homologous pairs. So they're lining up in tetrads next to each other. So if they all line up with their homologous pairs, you're actually going to only have 23 pairs deep. Does that make sense? You'd only have 23 pairs that line up here. So what ends up happening is 23 chromosomes go this way, and 23 chromosomes go that way. Now they are duplicated, but you still already cut your chromosome number in half. Now there's another thing that can happen here that leads to genetic variation in your offspring. And it's called independent assortment. Which side each chromosome lines up on is independent of the other pair. So let's say the blue are your paternal chromosomes and the red are your maternal chromosomes. Now, you can get so many different combinations here. You can have where, maybe I can draw it for you. What I mean by independent assortment is, let's say we had all the maternal line up on one side just by chance. I'll just do 3 chromosomes, and then all the paternal line up on this side. What that would result in is you would produce one gamete that has all paternal chromosomes, because these are all going to go this way, and one gamete that has all maternal chromosomes. Okay. Well, most likely you don't have all your mother and all your father's genes. You probably have a combination. So what independent assortment is that they can line up on different sides. So you could have, let's say, this combination. So let's say they line up this way, okay, because it just is random. So this cell will end up getting the paternal chromosome 1 and the maternal chromosome 2 and 3, and this cell will get a different combination. This egg or sperm would have a different combination. It would have the maternal chromosome 1 and the paternal chromosome 2 and 3. Now imagine all the different combinations I can make if I have 23 pairs of these. So what side they line up on is independent of each other. And then you separate them. So in anaphase 1, you're separating, like we said, the homologous chromosomes. And then you have telophase and cytokinesis. And what that results in is 2 haploid cells, because they only have 23 chromosomes, but the chromosomes are still duplicated into sisters. Then you go into meiosis 2. Prophase, I'm not going to talk much about here. If you remember from mitosis, the centrioles moved opposite sides. The mitotic spindles form, the nuclear membrane breaks down. Metaphase 2, how this is a little different than metaphase 1, is now all the chromosomes are going to line up vertically. So you'd have 23 of them line up, the 23 duplicated chromosomes line up in a line. And then in anaphase 2, you're separating the sister chromatids. So 23 sister chromatids go this way, and remember now we call them daughter chromosomes. Is it all coming back to you from mitosis? Okay. And then you have telophase 2 and cytokinesis, which results in 4 haploid gametes. So let's review. There's 3 factors that can contribute to genetic recombination. Literally, you can produce as many eggs and sperm as you want, and most likely you don't produce two eggs that are identical. If you do, that's just chance. But there's so many different genetic combinations that you can produce. And the reason has to do with these three factors that contribute to different genetic recombinations. So one is, remember we talked about crossing over, which can happen whenever the tetrads are together. So during prophase and metaphase 1. So remember, that's when you kind of swap pieces of chromosomes. So if you're just looking at crossing over, if you can imagine this looking at just one pair and you have 23 pairs, and say I swap some DNA here and here, and then in metaphase 1 I separate my homologous pairs, so I have a sister that's still completely paternal, and I have a sister that has some maternal and paternal genes. Same here, some maternal and paternal genes, and one chromosome that's completely still maternal. So what will end up happening is I end up with a gamete that has the paternal chromosome 1, the maternal chromosome 1, and then two gametes, two sperm or two eggs that have a combination chromosome. That's a little of each. So we call that a recombinant chromosome. That can lead to 4 different offspring, that genetic combinations. Then we talked about independent assortment. This can also lead to more genetic recombination. And this happens in metaphase 1 and 2 because it basically depends on what side the chromosomes line up, what cell it goes into. So this is just showing 2 different possibilities. Let's say we're looking at just 2 pairs and you actually have 23 pairs. They can line up like this, or they can line up like this where they're on opposite sides. So in this case, you produce 2 cells that have, say this is chromosome 1 and 2. They'd have the paternal chromosome 1 and 2, and then you can produce 2 gametes that have maternal chromosomes 1 and 2. Or if they line up like this, you can end up with gametes that have paternal chromosome 1, maternal chromosome 2, or they could have maternal chromosome 1, paternal chromosome 2. Okay, but you're basically seeing you're getting 4 different combinations of gametes. Now, you can actually calculate how many combinations you can make based on independent assortment. It's just basic probability. There's 2 possible sides that the chromosomes can line up on. Raise it to the power of the number of chromosomes you have, which is 23. If you do 2 to the 23rd, there's 8 million different combinations of genes that can be put into sperm or egg. So just based on independent assortment alone, I can make 8 million different eggs that are all genetically different. Now combine that with what we call random fertilization. The egg is going to be randomly fertilized by one sperm. So if as a female I can make 8 million different combinations based on independent assortment, and I release one egg, which is random, the egg that I release is random, and the sperm that gets to that egg is random, and there's 8 million different combinations of sperm that males can make. If you multiply those together, there's actually 64 trillion possibilities, genetic possibilities for your offspring. So that means if 2 mates produce 64 trillion offspring, there's a chance that none of them are genetically identical. Okay, now that's not going to happen, but it's just to blow your mind about how we could all produce our offspring that none of them are going to be genetically identical to each other. And that's why you do not look identical to your sibling unless you're an identical twin. You might look pretty similar to your sibling, and that just is luck that you probably got a lot of the same genes, and that's possible too. And this number isn't even considering crossing over, which we can't really calculate the rate of crossing over. And so there's actually a bigger possibility there. So that's just to shock you a bit, but I'm sure you kind of realize that. Now sometimes students ask me what the difference between an identical twin is and a fraternal twin. Some of you might already know this, but identical twins come from the same fertilized egg. So one egg was fertilized, and then the cells split after fertilization and then develop into two separate offspring. And so genetically they are identical. Now fraternal twins, some females will ovulate 2 eggs a month. Some could ovulate 3 and you can get fraternal triplets. But they're 2 separate eggs that were fertilized by 2 separate sperm. So because all of our gametes are different, fraternal twins aren't genetically identical. And that's why you can get a male-female twin. Alright, so let's look a little more at gametogenesis. So you produce your gametes through meiosis. And it begins in utero and it will resume during puberty. And we'll talk about the differences in that between male and female. First, you have to produce the stem cells in your gonads. So when you were an embryo, the cells that developed into your ovaries or testes, they underwent mitosis to produce the stem cells. And the stem cells are called spermatogonia in males and oogonia in females. Then those stem cells start to undergo meiosis when it's time to produce gametes. And when they undergo their DNA replication, which would happen during interphase of meiosis, specifically the S phase, remember the S phase? Then once you undergo DNA replication, we call that gamete the primary gamete. So it has 46 duplicated chromosomes at that time. Then it undergoes meiosis 1. So after interphase, it undergoes meiosis 1, and you end up with 2 cells that have 23 duplicated chromosomes. You call those secondary gametes. And then you undergo meiosis 2, which gives you your 23 chromosomes, haploid cells, and that we would call your final egg or spermatid. Spermatid is a term for a sperm before he can swim. So let's look at this picture. This is showing the gametogenesis because it's a little different between males and females. So you start with your stem cells, your spermatogonium or spermatogonia if you're talking about plural, spermatogonium if you're talking about one, if you're male, and oogonia if you're talking about it in females. So these are the stem cells. So they are diploid and they are produced through mitosis, normal cell division. In males, you're producing all the stem cells while you're an embryo, and it will continue when you go through puberty. In females, this happens while you're an embryo and you're developing. So let's stick to males first. Let me organize my thoughts here. Let's start with males because they're actually easier to explain. So you have all the stem cells, the spermatogonia, which would be in the testes, and then when you start puberty, these spermatogonia are going to start to undergo meiosis to produce sperm. And the first thing you're going to do is produce your primary spermatocyte, which remember primary is when the DNA replicates. Okay, so you replicate your chromosomes into sisters. Then you undergo your first mitotic division, and that will result in 2 haploid cells, but your chromosomes are still duplicated. So that's meiosis 1. And then those cells will divide in meiosis 2 to produce 4 gametes that have 23 chromosomes and are haploid, one set of each chromosome. Now in males, this process is continuous. When a spermatogonia decides to undergo meiosis, it's a continuous process. It happens continuously. And in males, they're producing a large amount of sperm per minute. Now females is a little more complicated, and it's kind of interesting. In females, what happens when you're developing, you produce all your primary oocytes. So the oogonia will undergo DNA replication for S phase and duplicate all your chromosomes. And you have all the primary oocytes you're going to have before you're born. And then what happens is they're arrested in that state until you reach puberty, and then the meiosis will pick up. And in meiosis 1, same thing, you're producing your secondary oocytes. Now what happens in females is when the cell division occurs, the cytoplasm isn't divided evenly. So you end up with what is called a polar body that breaks down. So only one cell is produced, one egg is produced, one secondary oocyte, and then the other half of the chromosomes go into a smaller cell that degenerates, okay, or disintegrates, I should say. This secondary oocyte is what you actually ovulate. So this is what's very interesting. You ovulate an egg that hasn't undergone meiosis 2 yet. It doesn't undergo meiosis 2 until fertilization actually occurs. So when the cell is fertilized, what will happen is the egg will spit out the other half of the DNA, and it will become your second polar body, and it breaks down. And then your 23 chromosomes and your 23 chromosomes from the sperm. It's a little weird. Females will arrest in what we call metaphase 2, and the cell just gets arrested there until fertilization occurs. And it's arrested as a primary oocyte basically from when you're an embryo until you reach puberty, or you might not ovulate that egg until you're 40, right? So it could be arrested in that state for 40 years. This is what they think is linked to certain trisomies like Down syndrome, because Down syndrome is when you end up with 3 21st chromosomes. And so what they believe happens is these cells are arrested, and it's called non-disjunction. And the proteins that hold the chromosomes, your mitotic spindle, as they think as it ages, it becomes less efficient at separating the chromosomes properly. These chromosomes don't separate properly, and so it stays, and you can end up with 1 21st chromosome from the sperm, one from the egg, and the other sister that never got pulled apart from it. And so you end up with 3 instead of the 2 that you should end up with. And that's why your chances of having a child with Down syndrome increases as the mother's age. And they've also seen it even with the fathers, because non-disjunction can happen in sperm also. Alright, so spermatogenesis, the production of the sperm, begins at puberty and continues throughout life for males. And where it's occurring, where these cell divisions are occurring, is in the seminiferous tubules of the testes. If this was a cross-section of a seminiferous tubule, this would be the lumen of the tubule here, and this would be where your spermatogonium, the stem cells, are, and they're undergoing meiosis 1 and 2 to produce the sperm, and those gametes will be put into the lumen of the tubule and eventually go to the epididymis, and then through the vas deferens to be ejected. Now, spermatogenesis is controlled by hormones. Guess what? Hormones. So I kind of outlined it here, and you can read that on your outline or in the PowerPoint, but it's easier for me to explain it using this picture. So you have it there in text, and here I'm going to explain it in a picture. So what triggers sperm production? We have a hormone called gonadotropin-releasing hormone that is produced by the hypothalamus. And that hormone causes the anterior pituitary gland to produce FSH and LH. Now this picture is deceiving because it looks like they're trying to show the posterior pituitary gland producing the LH, but the LH is also produced by the anterior, and they don't have a little arrow. I'm going to draw an arrow in here because gonadotropin releasing hormone triggers the release of both FSH and LH from the anterior pituitary. FSH is going to target the cells of the testes and play a role in follicle development, basically production of the gametes and cell division. And then LH targets the Leydig cells and causes them to produce testosterone. Now, testosterone plays a role in the maturation of of the sperm, but they also play a role in giving you your secondary male characteristics. Now, as in all hormones, there's a negative feedback loop because you don't want to just keep producing gonadotropin releasing hormone and continually producing FSH and LH. You want to keep everything in balance. So there's a hormone called inhibin that inhibits the production of FSH once FSH gets to a certain level, and testosterone will shut off its own production by inhibiting gonadotropin releasing hormone and LH. See, did I cover everything on this slide? So LH will target Leydig cells, produce testosterone, which gives you your secondary male sex characteristics. And then FSH will target the Sertoli cells and play a role in spermatocyte maturation. Now females, we call it oogenesis, the production of the ova, and it's produced by meiosis. And then we already talked about the total supply of primary follicles are present at birth, and then your ability to ovulate the eggs begins at puberty. And you can see you have your primary follicles that will develop into your secondary, tertiary. You eventually have your mature follicle, and then you'll ovulate the oocyte from that mature follicle. Most females ovulate one egg a month. Okay, you usually alternate the ovaries. But there is some females that will ovulate an egg from each ovary. Some females that ovulate every other month. It just depends. But most ovulate one a month. So what we're going to talk about in females, the hormones are a lot more complicated in females. And we're going to talk about changes that happen. You all know that females have a monthly cycle that occurs. And we call this the menstrual cycle. And there's cyclic changes that occur over and over each month. And there's changes that occur in the ovaries, which we call the ovarian cycle. And then there's changes that are occurring simultaneously in the uterus, called the uterine cycle. So we're going to go through what's happening in the ovaries first, in the ovarian cycle, and then we'll go over the uterine cycle, the changes that are happening in the uterus. So there's kind of three phases of the ovarian cycle. There's the follicular phase, ovulation, and then the luteal phase. So what's happening during the follicular phase is the follicle is developing in the ovary. So there's actually a few primary follicles each month that will start to develop, but one of them will become the dominant follicle, and the others will disintegrate. And so the primary follicles will start to develop and become secondary follicles and mature follicles. You have this fluid building up in the antrum, and then what's triggering that follicle to develop is that follicle is expressing, as it's maturing, more and more receptors for FSH. So it increases its sensitivity for FSH. So you don't have to secrete very much FSH because it's very sensitive to it. And it's also producing an increased amount of estrogen. Okay, the ovaries will be producing estrogen, and that matures the follicle. Okay, so during the follicular phase, the follicle is growing. That's easy to remember, right? And then ovulation is just ovulation. We're all pretty familiar with that. That's when the ova is released from the ovary. What triggers the ovulation is a surge in the hormone LH, which is your luteinizing hormone. And then that ruptured follicle, the cells of the ruptured follicle, will become what we call the corpus luteum. That means yellow body. Corpus means body. Luteum refers to yellow. So it's the yellow body. And these cells will then start to produce estrogen and a high amount of progesterone, the hormone progesterone. And these hormones are going to help prepare the uterine lining for pregnancy, or if that egg gets fertilized. So let's look at a diagram of these hormones and what's happening in the ovarian cycle. So during the follicular phase, you have your follicle developing here. It's maturing. And remember I said LH is pretty low at this time. And I told you that FSH, you have a slight increase in FSH, but it's very little. And that's because you don't really need very much because these cells are becoming more and more sensitive to the FSH. But what you'll see is during this follicular phase, estrogen levels are climbing as the follicle matures. And it's these rises in estrogen levels that then cause a spike in FSH and LH. Okay, and this is around day 14 approximately where you have ovulation, because it's this spike in LH that triggers the ovulation. And then those levels will drop off, and your corpus luteum will start producing estrogen. So you have a kind of another spike in estrogen, a smaller spike, but you have an estrogen stays relatively high. And you can see that progesterone, which was very low during the follicular phase and ovulation, starts to increase quite a bit. This is during the luteal phase. Now what will happen is, if fertilization doesn't occur, the corpus luteum dies off. And it becomes what we call the corpus albicans, which is basically scar tissue. So albicans refers to white, so now it's a white body. And these hormone levels, the estrogen and the progesterone, will drop off. So that's what I was just kind of talking about. The corpus luteum will produce increasing amounts of progesterone and estrogen for about 2 weeks. And so if fertilization does not occur, then the corpus luteum turns to scar tissue and stops producing the hormones, and the hormones drop off, and that drop in progesterone and estrogen triggers menstruation. If fertilization does occur, the cells of the fertilized egg will start to secrete the hormone HCG, or human chorionic gonadotropin hormone. And this hormone causes the corpus luteum to persist and stick around. And so the corpus luteum will keep producing progesterone and estrogen, and then those levels will stay high and not drop off, so menstruation will not occur because you never have that drop in hormone. And the corpus luteum will continue to produce progesterone until about 10 weeks gestation. Okay, so if you have fertilization, it's until the embryo's been developing for about 10 weeks that the corpus luteum will keep your progesterone levels high. And at that point, about 10 weeks, is then when the placenta has developed enough to take over the hormone production from the corpus luteum. All right, let's look at the uterine cycle. What's happening in the uterus during the monthly cycle? And this has to do with changes in the endometrial lining of the uterus. So if you review your anatomy of the uterine wall, you have, connective tissue on the outside. Then the uterus is made up of a lot of smooth muscle, which is the myometrium. That's the smooth muscle portion, because we all know the uterus can contract. That's what happens during labor. And then the inner layer lining the lumen of the uterus is called the endometrium. And this is what changes during your monthly cycle and gets shed. It's a cycle, so you just got to pick somewhere to start. So we're going to start with menses, which is the shedding of the lining, or menstruation, which happens once a month. And this is triggered by that drop in progesterone and estrogen if fertilization of the egg does not occur. And so in that case, you're shedding the endometrium. Then what happens is the proliferation phase, which is after the endometrium is shed. You're going to need to rebuild it to prepare for if pregnancy occurs next month. Okay. So what will happen is, under an increase in estrogen levels, the endometrial lining is going to divide and produce more cells and add new layers. Then there's the secretory phase, which is where you have an increased blood flow to the endometrium. So you have these little arteries or capillaries that start to grow into the endometrial lining. You have increased blood vascular supply to the cells, and increased glandular secretions. It's a lot of mucus production. And that's under the influence of the increase in progesterone. So if we look at this graph again, now down here, you can see how the uterine cycle coincides with what's happening in the ovary, and the ovarian hormones, so these are estrogen and progesterone produced by the ovaries. Gonadotropic hormones are released by the pituitary gland, your FSH and LH. So let's put this all together. So you just started day 1, which we usually mark as the first day of the menstrual cycle, and you're shedding your menstrual lining. And at this time, what triggered the shedding of the lining is this drop in progesterone and estrogen. So you're shedding the lining, and then what's happening is, remember, your estrogen levels start to rise during the follicular phase, and that rise in estrogen levels cause the cells of the endometrial lining to start to divide and proliferate. Okay, so it's thickening the layer. And then what happens is you enter into the secretory phase, which coincides with the luteal phase of the ovarian cycle. And this is when your progesterone levels are high. And when the progesterone levels are high, it increases the vascular tissue, increases the blood supply to the endometrial lining, increases mucus production, and that's the secretory phase. And then the cycle occurs. If fertilization doesn't occur, the corpus luteum dies, and the progesterone levels drop, and it triggers menses, and you start all over again. So here's a chart of the hormone control of the menstrual cycle. Like I said, it's pretty complicated, but we'll just try to keep it simple. So if you look at early to mid follicular phase. So gonadotropin releasing hormone causes FSH and LH to be produced, but it's pretty low levels. You can see that there's a negative feedback here. It's pretty low levels. And you start to produce the granulosa cells of the follicles start producing increasing amounts of estrogen. And that's why you start to see this increase in estrogen here. And estrogen works as a positive feedback mechanism on itself. It causes it to produce more and more estrogen. Now when estrogen gets really high, or starts to get pretty high, estrogen's what's confusing, because estrogen kind of in low levels works as a negative feedback. At higher levels, it works as a positive feedback, and if it gets too high, along with progesterone, it works again as a negative feedback. So it's really tricky with the estrogen. But as estrogen increases, it starts to work as a positive feedback mechanism for the gonadotropin releasing hormone, which will cause the FSH and the LH to spike here. And then what happens is in the luteal phase, so then you have ovulation, then you enter the luteal phase, and your estrogen and your progesterone start to increase. And the combination of really high progesterone and somewhat high estrogen then goes back to working as a negative feedback on the gonadotropin releasing hormones. So your FSH and LH will drop back off, okay, and it's your corpus luteum that are secreting these ovarian hormones. And then your late luteal phase, your estrogen and progesterone levels drop because the corpus luteum dies. That starts the cycle over again, where the gonadotropin releasing hormone will increase the FSH and LH slightly, and the new follicles will begin. Alright, so this is just a little picture of what happens during fertilization. Remember I talked about how the egg is actually arrested in metaphase 2. So you have 23 of the sisters here, 23 here, and then fertilization occurs, and when that sperm nucleus enters into the egg, the egg will basically spit out half of the chromosomes. It will finish its meiosis 2, and you get your second polar body, and then the 23 remain in the egg. So that's just kind of interesting that that happens at fertilization. And now you have your zygote. Ovulation, and then you have fertilization, and fertilization usually occurs within 24 to 48 hours of ovulation. And then the cells will start to undergo mitosis right away, start dividing as it travels down the fallopian tube. And by day 4 or 5 after fertilization, you have what we call a blastocyte. And a blastocyte has an inner mass of cells, and these are the cells that are going to basically become the baby, or the embryo. And then these outer cells here are called the trophoblast, and these are the cells that will eventually become, later on, the placenta and the umbilical cord. And the blastocyte will implant. It's about 7 days that the blastocyte reaches the uterus and will implant, and that's all triggered by chemical signals. Prostaglandins will allow the blastocyte to basically bury itself, where it buries itself and it lives off the endometrial lining until the umbilical cord can develop and the placenta and the umbilical cord can develop. And as I said, the placenta, after about 10 weeks after gestation, will start taking over hormone production for the corpus luteum, and then the corpus luteum will disintegrate. Alright, so that is reproduction. We haven't gotten into labor and delivery because I don't want to completely overwhelm you on your final. But if you're interested in looking at that, your book does cover labor and delivery briefly. But this is kind of stuff that I wanted to get through that you might not be as familiar with, such as sex differentiation with the bipotential structures. And then, of course, in physiology, we got to get into all those complicated hormones and negative feedback loops. So that is it. That is the end of lecture for good. You are done, and now you just need to study for that final, and that is it. So thank you for a great semester, and I wish you the best of luck.